time for. So we may skip some of the questions like we've been doing. I'm still working out the timing of this new book stuff. So do get logged in. Homework for this chapter will be due on Wednesday, and I'll remind you of that in a couple of days. Okay, so last time we ended up talking about the stomach, and then we talked about the small intestine and all the things that it does. And so we have all these different areas of the digestive tract, and they're all doing different things. So whenever you have that condition, you have to have some way of controlling and managing these different pieces. And early on, so in the first lecture on this topic, we talked about the different layers of control. You know, that local factors play the largest role. So essentially, the digestive tract manages itself, but then also that there's neural controls and hormonal controls. So the 5 hormones that I want you to know about are all listed on this one diagram, which is right from your book. So there are dozens of hormones that play a role in the digestive tract, but we're only going to focus on these 5 because in some ways these play the largest role in in managing the digestive tract the way that we're looking at it. Okay, so the first one is gastrin. So gastrin, when you think gastrin, think stomach. Gastrin stimulates the stomach to do what the stomach does, which is to produce acid and produce pepsinogen and churn. In other words, move and bend and contract to mix things up that are inside it. So gastrin has 2 principal sources. One is the stomach itself. So the G cells that we looked at in the stomach glands, they produce gastrin, and gastrin is also produced by the duodenum to some extent. But when you think gastrin, think stomach. All right, so that's that one. Secretin, not surprisingly from its name, it triggers the secretion of stuff into the GI tract, namely bile from the liver and pancreatic juices, or what the pancreas produces and secretes into the GI tract. So we have bile and buffers, your book calls it. So what's a buffer? A buffer is a substance that can either neutralize acid or protect the pH. In other words, keep a pH stable. So secretin stimulates the production of those 2 things. And we're going to end today's talk by talking about the accessory organs, the salivary glands, the liver and the pancreas, and the gallbladder. So secretin very much operates on those accessory glands, on the liver and on the pancreas in particular. All right. And secretin also reduces gastric activity. One of the things you'll see is the duodenum, being the first part of the small intestine, it has to control the stomach because the stomach can only empty as fast as the intestine can handle what's coming in. So the intestine is often telling the stomach to slow down, and secretin is one of those hormones that does that. All right, so that's secretin. GIP, gastric inhibitory peptide. Well, it kind of tells you what it does. Gastrum is stomach, so this is a peptide, which just tells you what kind of a hormone it is, that inhibits the stomach. So when fats or high amounts of carbohydrates enter the duodenum, that first part of the small intestine, gastric inhibitory peptide is released, and it slows the stomach down. Now, the reason for that is fats take a long time to digest. So the digestive tract has to slow down in order for fat digestion to complete before the substances enter the large intestine to be gotten rid of. And so slowing the stomach down slows the whole system down to give time for those fats to digest. So that's one. What about the carbohydrates? Carbohydrates are absorbed very rapidly. So in order to prevent a big spike in blood sugar after eating a meal that's high in carbohydrates, there again, the gastric inhibitory peptide slows everything down so that that sugar comes into the small intestine more slowly, so that there isn't a big spike in blood sugar, but instead it's sort of a slow and steady instead. So does what it says, gastric inhibitory peptide. All right. CCK, cholecystokinin. I always think of this as the fat hormone. In other words, whenever there's lipids present in the duodenum, in other words, a meal high in fat, so McDonald's, anything practically, CCK is released, and its targets are really two. One is on the liver and the gallbladder, and that is to increase the production of bile. And then the other target is at the pancreas. The pancreas is what produces the enzymes that actually digest fats. So in order to digest fats, we need bile and we need pancreatic enzymes. And CCK stimulates both of those things. And because fats take a long time to digest, CCK also inhibits the release of gastrin. So just like GIP, cholecystokinin, slows down the digestive system to give a chance for those fats to get digested. All right, so CCK. And then the last one is VIP, vasoactive intestinal peptide. It is also released from the duodenum, and it stimulates blood flow. Ultimately, we want to absorb these nutrients that are coming in through the digestive tract. In order to do that, we need to have a blood supply. So VIP encourages blood flow through the areas of the intestine that are actively absorbing. All right. So of these 5, there are 3 that I think are extra important, and that's gastrin, secretin, and cholecystokinin. So if you can mark those, you're likely to see those 3, on exams, basically, because those are the big 3, gastrin and cholecystokinin for sure, and the secretin is sort of a third. These, the GIP and the VIP, they're important and they're playing a larger role today in the treatment of things like diabetes, but their roles are not as clear. All right, so here's gastrin. Gastrin stimulates the stomach, should produce acid, and then motility. Secretin and CCK both kind of do the same thing, and that is to get the pancreas to do its job, get the liver to produce bile. Bile is necessary to digest fats as well, and that's because, as we'll talk more about in the next chapter, we all know that oil and water don't mix, right? Well, bile contains these substances called bile salts, which essentially are like detergent, like non-dish soap. So when you mix soap with oil and water, you can actually get the two to mix. So we need to have the bile from the liver in order to make that happen. All right, so secretin and CCK. And so we have inhibition and we have stimulation. So gastric inhibitory peptide, the way it works is it blocks the release of gastrin. So this is just sort of a concept diagram of what we were just talking about. All right, so the hormones. Okay, so as we eat a meal, the stomach fills up, right? And then eventually, when we're done eating the meal and we're living our daily lives, the meal that we ate is slowly released into the intestine for digestion and absorption. So the stomach plays sort of a critical role in the control of the digestive process, of the process of consuming and then absorbing the nutrients from a meal. So because of that, we divide the stomach's activities into 3 phases associated with eating a meal. The first phase is called the cephalic phase. So cephalus is head. So this is the phase that comes from the head. So what do we mean by that? In order to have the stomach be as effective as possible, it kind of needs to be turned on before it's needed. You know, just like a car in the dead of winter has to sort of warm up before it can perform correctly, the stomach is a little like that too. So ideally, we want to have the stomach ready for food before food is actually in it, okay? So we call this the cephalic phase because it's directed by the central nervous system. So when we are thinking of food, smelling food, seeing food, preparing for food, you know, when we know that a meal is coming, automatically the central nervous system starts to prepare the stomach for that meal. So like, when we've all experienced this, it's about time for dinner and your stomach starts growling, so to speak. That's the cephalic phase at work. And it all happens through the vagus nerve, which is one of the principal autonomic nerves that come out of the brain. So in response to those concepts of getting ready to eat, the stomach is stimulated to produce mucus, acid, and enzymes, namely pepsinogen. So we get, once we have those three things, well, now the stomach is ready to go. When that first bit of food arrives at the stomach, the process of digestion can already begin, okay? So that's the cephalic phase. What follows is then the gastric phase. So as we eat, with every bite that we take, the stomach relaxes a little. We talked about how the stomach can contract to be relatively small, and then it can relax to get much, much larger to hold more food. Well, as we eat, that relaxation occurs, and we get some distension. As we put food in the stomach gets stretched out a little. And its response to that distension is relaxation. So the more we put in, the bigger the stomach gets until the stomach reaches its maximum size, right? All right, so that's one thing that's happening, is that relaxation. And then another is once food is present in the stomach, the presence of that food is a stimulus to produce acid, pepsinogen, and mucus. So in the cephalic phase, we've got an empty stomach, right? So there's no stimulus to make those three things. But once we start putting stuff in the stomach, that stuff becomes its own stimulus. So in the gastric phase, instead of the vagus nerve triggering the production of mucus, pepsinogen, and HCl, the contents itself are doing that. So we get mucus, pepsinogen, and HCl, all in response, or all because of the presence of stuff in the gastric, or in the stomach itself. Okay. So the meal is over, and the contents of the stomach have been processed. All right. So the pepsinogen, which becomes pepsin, when exposed to acid, the pepsin has done its protein digestion work. The acid has done its work. The mucus has protected the lining of the stomach. So now it's ready for now it's time for the contents of the stomach to then move on to the rest of the system. So we call that the intestinal phase. And this, another name for intestinal phase, you could call it gastric emptying. In other words, the stomach got filled up, and now it's got to empty into the small intestine in order for the rest of the work to get done. All right? So the intestinal phase is controlled by the duodenum. So when the duodenum is ready to receive some chyme from the stomach, it sends a signal that causes the pyloric sphincter to relax and the stomach to contract. So a small amount of chyme comes out into the duodenum, and then that sphincter closes again. So it's a little bit at a time. The stomach can hold a very large amount, but the duodenum can only hold a much smaller amount. So all it can do is take a little at a time, process it, and then move it on, and then that cycle repeats. All right, so in that intestinal phase, the duodenum controls the emptying of the stomach, so that the rest of the processes of digestion and absorption can happen. All right, so cephalic, gastric, and intestinal phases of the stomach. All right. Okay. So, what happens after digestion and absorption have occurred? And like I said, we're going to talk more about those two pieces in the next chapter. But so by the time substances reach the ileum, the end of the ileum is the end of the small intestine, right? And by then, everything the body wants to get out of that what was previously food, that chyme, everything has been absorbed and digested already. So what enters the large intestine at the ileocecal junction is stuff the body needs to get rid of or recover the fluid and salts from. All right, so the large intestine does that role. It reabsorbs fluid and it compacts the feces so that of the food that we eat, only a small amount of the undigestible material is then ejected out the other end. All right, so our anatomy here is important for the large intestine. All right, so we start here. Here's the ileum. The large intestine starts with a kind of sac-like area called the cecum. So this is all cecum right here. Hanging off the bottom of the cecum is the appendix, which we've all heard about. The appendix in the human is considered vestigial. In other words, it's not an active organ in us, so it can be taken out without any downside. Now, the truth is, the appendix is actually a lymphoid organ, like we talked briefly about in another chapter. In other words, there are a bunch of immune system cells that live in the appendix. And in other animals, it's very, very long, the appendix is, like 3 or 4 times as long as ours is. So it's probably a way to control the bacterial population that lives in the large intestine. So, does it have a role? Probably. But in humans, you don't have any downside for having had an appendix taken out. All right, so that's the cecum. Where the ileum joins the cecum, there's this little sort of puckered valve that you see right here. That's called the ileocecal valve. So ileum, cecum, ileocecal valve. And it's important, it's closed most of the time, and it only opens when substances leave the ileum and then start their journey through the large intestine. So just like the stomach, the pyloric sphincter is closed all the time, opens to allow some emptying and then closes again. The ileocecal valve works in that same way. It opens, substances enter the large intestine, and then it closes again. And that's useful because, remember, the large intestine is not sterile. There's a bacterial population that live in the large intestine, whereas what comes out of the ileum is mostly sterile. So by having this valve here, it helps to prevent the bacteria from flowing sort of backwards through the system. All right. So once we get into the cecum, now we get into the colon. Colon is just another name for large intestine. They mean the same thing. All right. So first, the colon goes up. So we have the ascending colon going up. Then it goes across. We call this the transverse colon. Transverse just means to go across. That first bend where we go from up to across, we call that the right colic flexure. A flexure is a bend. Colic is having to do with the colon, and obviously this one's on the right. Another name for this is the hepatic flexure, and that's because the liver sits right here. So right next to the liver, we have the right colic flexure, also called the hepatic flexure. All right. So then we go across, and then that transverse colon now goes down, and where it makes the bend, this is the left colic flexure, also called the splenic flexure, because the spleen sits right about here. So we have a liver flexure and a spleen flexure, so right colic and left colic flexures. All right, so transverse, then descending. Then the descending makes this kind of S shape right here. You can see that S. That's the sigmoid colon. And then the sigmoid colon ends in the rectum. And the rectum you can usually tell because its walls are different. In the whole path of the large intestine, the anatomy of the tube itself is pretty consistent. So we have these little pouches, like here's one, here's one, here's one. Those little pouches are called haustra. So a haustra is a pouch. I'm sure that word is on here somewhere. Yeah, here we go. So these little pouches are haustra. And then running along the top and bottom of the large intestine is this band of smooth muscle. You see it here and all the way down here. That's called the tinea coli. And it's all that's left. When we looked at the small intestine and the stomach, we saw that there were 2 layers of smooth muscle, right? Well, the large intestine has 2 layers too, but the tinea is all that's left of the outermost layer. So instead of having a complete covering of 2 layers of muscle, there's a circular covering, but then the tinea is all that's left of that outer longitudinal layer. All right, so let's see, I think that's the anatomy I needed you to know here. So hanging off of the colon are these little fatty deposits right here. Some people have more than others. And the whole large intestine has a blood supply that comes from the aorta and then returns to the portal vein, which we'll talk more about in a different chapter. All right. So up, across, down, around, and out, right? So ascending, transverse, descending, sigmoid is the around, and then rectum is the down and out. All right, so do know this anatomy. Okay. So what's it for? In order to accomplish digestion and absorption, the body has had to put a lot of fluids into the GI tract. So, let's say that, you've eaten a piece of toast. Well, the toast doesn't have very much fluid in it. But in order to digest and absorb that, the body has to put fluid in to make that happen. It starts even in the mouth. When you eat that toast, your salivary glands work. Well, that's the body putting fluid in. The large intestine is present to take that fluid back out so that what we've put in to make digestion and absorption happen, we can recover. Particularly fluid and electrolytes. So the 3 important things about the large intestine are fluid, electrolytes, and bacteria, right? So because the fluid is going to get reabsorbed, electrolytes are going to get reabsorbed, and the large intestine is one of the few places in the body that is not sterile. So we have a bacterial population that lives there as well. More on that in a second. Okay. So if we take a look at the wall of the large intestine, we can see that unlike the small intestine, the wall is smooth. In the small intestine, we had lots of folds, right? Folds of folds of folds. Well, the large intestine's job is different. It doesn't have to absorb nutrients anyway. It's going to reabsorb water. But it does have to protect itself. By the time stuff enters the large intestine, it's pretty nasty, from the body's point of view. It's all stuff that the body couldn't digest, couldn't absorb, so it's caustic and it's damaging to tissues. So the way the large intestine evolved to handle that is it's got this smooth, flat surface, and then all the cells live underneath that surface. So as the feces pass by the top, that environment doesn't affect these epithelial cells as much. So similar in architecture, I think, to the stomach. The stomach had sort of a thick mucosa with all these little grooves and pits, and so does the large intestine. So we have these intestinal glands. They do secrete mucus, which covers this surface, and helps to protect the underlying tissues from damage, right? So we have our same layers. We have the mucosal layer, submucosa, and then the muscularis externa. All right. So, and then the large intestine ends in the rectum, and the rectum has a couple of unique features. One is its wall is very muscular, and it's because it's the rectum that's going to actually push feces out. So it has to have some strength to it. The rectum ends at the anal canal, right? So here's the rectum up here, here's the anus, and then the anal canal connects the two. And we have two bundles of muscle at that canal. We have an internal sphincter and an external sphincter. And what we're going to see is that the two work together to control the defecation reflex. It's the very same thing we're going to see in the urinary system. And that is, so it's a common system. The internal sphincter is involuntary, right? So it's controlling whether the organism needs to defecate or needs to urinate. The external sphincter, which is out here, is under conscious control. So it allows the organism to control when and where that elimination occurs. All right, so the rectum is normally empty. It fills up by what are called mass movements. The large intestine moves a little differently than the small intestine does. You know, the small intestine is doing that segmentation and peristalsis all the time. Well, when the large intestine moves, it tends to move in larger amounts. So a mass movement might move all the material from here to the transverse colon, all the material from the transverse down. So it sort of shuffles all this material in one direction. And then at the end here, the sigmoid colon, in response to a mass movement, all the contents of it will appear in the rectum, and then we have the defecation reflex that's triggered. All right, so the rectum is normally smooth, two sphincters, internal, which is involuntary, and external, which is voluntary. So more on that in a minute. All right. Okay, so what does the large intestine do? So fluid, electrolytes, and bacteria. Those were the three things I said, right? So what enters the large intestine is about 1,500 milliliters of material. What exits through the rectum is about 200 mLs of feces. So that is 1,300 mLs of fluid and electrolytes that have been recovered. And that is the large intestine's job, is to do just that. So you can sort of see what your large intestine is doing. If you've ever had a bout of diarrhea, which most people have, that's the kind of thing that happens when the large intestine isn't working the way it should. So all that fluid that's going into the digestive tract, instead of being reabsorbed and recovered, it's lost. So dehydration can occur very rapidly there. You know, your entire plasma volume is only about 6 liters. So if you lost 1,500 mLs for, an hour or a day, you would have lost your entire plasma volume in just 4 days. So the large intestine plays a very important role in recovering that fluid to prevent dehydration. All right, so that's one bit. Along with the fluid, we get electrolytes, which are recovered as well. And then the large intestine is colonized, at least after birth it is. It's colonized with multiple species of bacteria. And it's a symbiosis, we call it, which means both organisms benefit. The bacteria benefit because they have a constant food supply. You know, the things that we cannot digest and absorb, oftentimes bacterial species can. So they do. They live in the large intestine and they do that. And what they give us in return is 3 important vitamins. So one is vitamin K. Vitamin K is important in the blood clotting system. Vitamin B5 and biotin, which is another B vitamin, are necessary for various metabolic processes or steps. So as long as we're eating, the bacteria produce these vitamins, which we then absorb from the large intestine as well. So effectiveness of fluid recovery, and then the bacteria as well. All right. So what about that defecation reflex? Okay, so there's an involuntary component and there's a voluntary component. The involuntary component goes a little like this. You know, because of a mass movement from the sigmoid colon, the rectum gets filled. And that filling creates distension. And that distension triggers a stretch receptor. So in the walls here are neurons that sense stretch. So the first thing that happens is when that stretch occurs is that the internal sphincter relaxes. So the rectum fills, the internal sphincter relaxes, but then the external sphincter contracts. Because what we don't want, the organism wants to control the when and where of defecation for lots of reasons out there in the wild. So we need to have a process to allow the defecation to occur, but control the when and where. So we have this short reflex and the long reflex. The short reflex prepares the system to eject the feces. The long reflex coordinates the control of where and when. All right, so sigmoid colon or rectum gets stretched. That stimulates the nerves of the sigmoid colon and the rectum. We get increased peristalsis to fill that rectum and get ready to push it, the feces on out. The long reflex, if the brain gives the command to relax the external sphincter, then defecation occurs. If the brain does not give that command, then the system is sort of paused for a little while. And if that pause goes on for long enough, then the internal sphincter will close again, the rectum will relax, the sigmoid colon will relax. It's like a snooze button. The brain didn't say to defecate, so that reflex is put on hold. Now it's going to come back, obviously, and it's going to come that urge or that awareness of needing to defecate will come back when the next mass movement then fills the rectum even more. So eventually, the brain will order the relaxation of that external sphincter, and then defecation will occur. So a lot of similarities between this and what we'll see in the urinary system, because the micturition, which is urination, that reflex is controlled in kind of this same way. There's a short reflex and a long reflex. So what about in babies or in people with spinal cord injury? Their defecation reflex is all coordinated by this short loop. There is no long loop. So when the baby's rectum fills from a mass movement, the stretch receptors cause that increase in peristalsis, and that feces is ejected immediately because there is no long reflex to control the external sphincter yet. That's a learned behavior. All right. So, okay. We're going to skip these just in the interest of time because we got to get through this thing. Okay. So, that was the GI tract, the long muscular tube, right? The rest of the digestive system is what we call the accessory organs. So, these are organs that are going to put stuff into the GI tract. So we have the salivary glands up in the head, the gallbladder and liver here in the right upper quadrant, and then the pancreas, which sort of sits just nestled underneath the stomach. All right, the salivary glands are pretty straightforward. We have three pairs of salivary glands. We have the parotid, those are the biggest ones, so they sit just in front of the ear. If you kind of push on the side of your face where they are, you'll feel saliva come out into your mouth. And that's because the parotid glands have a duct that enters the mouth sort of midway back, just above the teeth. So that's the parotid gland, parotid duct. under the tongue, we have another pair of glands. That's the sublingual salivary gland. It opens with multiple ducts that are next to the frenulum of the tongue. And then we have the submandibular gland, which sits sort of tucked in underneath or just on the other side of the mandible on the bottom. And the submandibular gland also has a duct that empties into the bottom of the mouth, underneath the tongue. All three of these pairs produce the same thing, which is saliva. Saliva is 99.4% water, but what makes it interesting is the non-water parts. So saliva has some interesting characteristics. There are some proteins that are present in it to make it mucus-like. So it's actually slipperier than water. And it's a little thicker than water as well. So saliva has that. There are antibodies in saliva that help to fight off bacterial pathogens that might be in the food that we eat. There are buffers that help to control the pH of the mouth. So anybody who has salivary gland problems, usually they have teeth problems too. And it's because without these buffers, the bacteria that live in the mouth create acid, and the acid eats the teeth. So in patients who have chronic dry mouth conditions where they don't make enough saliva, their teeth are almost doomed to rot because there aren't these buffers and antibodies to control the bacterial population in the mouth. And then the saliva also produces a substance called lysozyme, and it is just plain and simple antibacterial. It's almost like soap in that it's very effective at destroying bacteria. All right. And then the last piece, which actually contributes to digestion, is called salivary amylase, which is a protein that an amylase breaks down carbohydrates. So, if you take a saltine or a cracker and you put it in your mouth, you don't chew it or anything like that, you just let it sit on your tongue, eventually it will literally fall apart. That is salivary amylase, which is breaking up the molecular structure of that carbohydrate, of that starch. And we're going to see how that works in the next chapter when we talk about carbohydrate digestion. All right, so 3 pairs of salivary glands. All right. And your book does note that the secretions of the 3 are not exactly identical, but for our purposes, I think it's fine if you know all 3 produce saliva. Saliva has this stuff in it. Okay, so I'm not interested in you knowing the difference. Okay. Okay, the liver. This is, we could talk for 2 weeks about the liver. So what the book has done, and I commend them for doing it this way, because most stuff about the liver does not come up for most healthcare providers. So it's good to know what the liver does, but a sort of basic understanding of that is really all most people are ever going to need. Because if you have big liver problems, you're going to need a liver expert anyway. So, we all have to know a little, but the complex stuff we can leave to other experts. All right, so the functions of the liver are long, right? So we got a whole bunch of them. So we'll look at just a few of these. Okay, so synthesis and secretion of bile. Bile is what the liver makes and puts into the digestive tract. And bile has multiple purposes, but one of its purposes is to help emulsify fats. Emulsification is what dish soap does in your dirty sink water, right? Water and oil separate. But if you take a detergent molecule and put it in there, it allows the oil droplets to form very tiny little droplets and effectively mix with the water. So in the body, those are called bile salts. And they're present, they are made by the liver and they're present in the bile. So the liver makes bile, stores it in the gallbladder, and then releases it into the small intestine at the duodenum. All right, so more on that later. The liver stores glycogen and lipid, which the liver is our metabolic manager. It tries to make sure that all the cells of the body have what they need all the time. Okay? And we're going to see more of that in the next chapter about how it does those things. All right. Let's see. The liver is it inactivates potentially harmful things. We're going to see that all of the blood that leaves the digestive tract, all of it passes through the liver before it can go anywhere else. So in the food we eat, there can be all kinds of things, right? There can be bacterial toxins, plant toxins, venoms even. So before those potentially dangerous chemicals get out to the cells of the body, the liver, it all has to pass through the liver first. And because the liver can do so many different metabolic things, what comes out of the liver is sort of filtered from compared to what went in. So toxins are inactivated, substances are completely broken down. It's one way to think of it is the liver is like the digestive system for the digestive system. So what the digestive system absorbs, the liver then takes apart and reorganizes for the body's own use. All right, and then some non-digestive functions. The liver is responsible for making most of the plasma proteins that we talked about early on in the course this semester. The liver makes the clotting factors that make blood clot. The liver destroys damaged or dying red blood cells. The liver can store some blood for use in, for mobilization and exercise, for example. And then the liver breaks down hormones and inactivates certain medications. All right, so lots of things that the liver does. Where is it? Well, the liver sits up here in the right upper quadrant. It sits next to the stomach, and it pretty much fills okay, so we have the right side upper fourth, so right upper quadrant is where we talk about it. And the liver fills up most of that space. It's the largest organ in the body. It weighs several pounds, and it's about that big. I mean, it's a very good sized organ. As you can see from our cross sections here, most of the abdominal space is taken up by the liver at that level, at this spot right here. All right. So its anatomy, for all of the liver is boring on the outside, complex on the inside. Its anatomy is fairly straightforward, but because liver cells can do so many things, it has this whole repertoire of activities, even though its anatomy is fairly simple. All right, so on the front surface, we have a right lobe and a left lobe. Note that they're not symmetric, right? The right lobe is quite a bit larger than the left lobe. Dividing the right and left is the falciform ligament, which has the round ligament at the bottom of it. The round ligament is what is left of the umbilical vein. So it actually goes all the way to the umbilicus, or all the way to the navel. All right. And then from the front, we see the gallbladder, its fundus, or its dome-shaped top, just sort of sticking out here from underneath. So right lobe, left lobe, falciform ligament. On the back, we can see the other two lobes. So we have the quadrate lobe, which sits next to the gallbladder, and we have the caudate lobe, which sits between the left and right, just to the left of the vena cava. So this is the caudate lobe right here. The coronary ligament, corona is crown, so the coronary ligament goes all the way around the liver here, where it comes up underneath the diaphragm. And then at the back side, we see the hilum of the liver. So we have the portal vein, the hepatic artery, and then we have the common bile duct that's coming out here as well. So that's this guy. And then hanging off that bile duct is the gallbladder, which is a sac that stores up bile when the digestive tract is sort of dormant, so between meals. The liver makes bile all the time, but it's only injected into the small intestine when it's needed. So when it's being made but not being used, it gets stored in the gallbladder. All right, so important anatomy of the liver. All right. So if we zoom in to look at the liver's structure, we can see that it's arranged in these sort of hexagons that are called lobules or sinusoids. All right. So we can see, we've got one side, two sides, three sides, four sides, five sides, six sides, right? So hexagon. And this one has all the pieces in it. So we have arteries and veins around the outside, and we have one vein in the middle. Okay, so blood comes into one of these from the outside. So at each corner, we have a branch of the portal vein, and we have a branch of the hepatic artery. So the portal vein is blood that's coming from the digestive tract, right? When we talked in the cardiovascular system, we looked at the hepatic portal system, where all the blood from the stomach, small intestine, large intestine, instead of just going back to the heart, it has to go through the liver first, right? And that path through the liver is through the portal vein. So this is blood coming from the GI tract. Well, these cells are living human cells, so they need oxygen too to do their thing. So oxygenated blood comes in through the hepatic artery. So we have red blood and blue blood that sort of mix here. Red from the lungs, blue from the digestive tract. And then through the center of each of these sinusoids, this is the drain. So just like a sink has a spout in one place and a drain in the other, the drain is called the central vein, and it's going to emerge from the liver as the hepatic vein. So portal vein and artery go in, they mix in these paths here called sinusoids, and then the blood exits through the center through the central vein. So, and then at each corner, in addition to the artery and vein, there's also this green thing. Now here, this is not a lymphatic, this is a bile duct. So the cells of the liver produce bile, and that bile is collected in this system of ducts called the bile duct. So while blood is flowing this way, towards the central vein, bile is flowing the opposite way. So it's flowing towards the corners, towards what are called the hepatic triad: artery, portal vein, and bile duct. So at each corner we have those three structures. So bile flows in the opposite direction. All right. So if we sort of zoom in even more and look, okay, so now we're at one corner, right? Here's the central vein, here's the hepatic triad. So we have blood from the portal system mixing with blood from the arterial system, so we see that here. The bile is collected through this tiny little network of tubules called canaliculi that connect to each cell. So you can see how there's sort of like a plumbing system here where the bile drains out. And then inside these paths between the artery and vein and the central vein are these cells that are called Kupffer cells. And Kupffer cells are like macrophages, and anything that doesn't belong in the blood that's passing by, they're going to snatch out of it and then destroy it. So, remember, what's coming from the GI tract, there could be all kinds of bad stuff in there. So in this way, the liver acts as kind of an immune system organ because it has these macrophages that are scanning for things that don't belong as the blood is passing from the hepatic vein through the central veins where it will go out into the rest of the body. All right, so Kupffer cells do that. They also store iron for use in red blood cells. And then heavy metals like tin, mercury, lead, gold, those kinds of things that don't really belong in our systems, the Kupffer cells hold on to those as well. So at least they sequester that material out of the rest of the circulation. All right. All right, and let's see, our last topic, because I know we're about out of time, is the gallbladder. We are out of time. Okay, so we'll pick up gallbladder next time, and we'll